Yeah. So in today's session, we have to talk about the coagulation of the blood. So let's look back from the beginning where we stopped. So the three steps in which how the blood flow is stopped is through vascular spasm, and the second one is formation of platelet plug, and the third one is called as coagulation. So if you have lost till where we have talked about, we have finished the entire details about what are the types of WBCs, their characteristic features, and when their numbers increase, etc. Okay, so please go through it so that you can follow it up very thoroughly. Yeah. So coming to homostasis, as I mentioned earlier, homostasis is homo is sorry, hemo is basically called as blood. Stasis is balance. So maintaining the blood balance becomes a very integral part for our human physiology. So now let's say that we have an injury. So whenever the injury happens, we know that platelets play a very important role in coagulation of blood or clotting of blood. So it's only one part of the story. But if you see the whole process, how the blood flow is stopped, it happens in three steps: vascular spasm, formation of platelet plug, and coagulation. Let's see each of them. What is vascular spasm? So you have a blood vessels. Okay, let's say it is a blood capillary, whether it is arterioles or venules. So once there is an injury, now what happens is the blood vessels are made up of muscle cells, smooth muscle cells. So there are two types of muscle cells. One is cylindrical muscle cells, and the other one is longitudinal muscle layers. Okay. So the cylindrical as well as the longitudinal layers, what happens is they help these blood vessels to withdraw from the site of injury. When I say that, so let's say the blood vessel is ruptured here, or this is where the injury is. So the blood vessel constricts or it becomes smaller so that the flow of blood is reduced. Okay. And it withdraws from its position. That means it moves backward. So for this, these muscle layers help this vascular spasm, preventing the loss of blood as well as infection. And the second one is formation of platelet plug. So this is where the platelets come into the role. So what do platelets do? So let's say that we have an injury. So if a blood vessel is injured, now what happens is the fibers that are present in this blood vessels, which we generally call it as collagenous fibers, as well as the innermost lining of this blood vessel gets exposed, right? Endothelial lining gets exposed. So when collagen and the endothelial lining is exposed when the injury happens, now that will make the platelets to become spiky and sticky. Usually platelets are very smooth, okay? They're very smooth. So now what happens is they start becoming spiky and they start becoming sticky. So each of the platelets, as they become spiky and sticky, they come together and form a plug-like structure. So this is called as platelet plug. Usually there are certain factors that monitor the rate of growth of the platelet. So for example, we have uh, Willebrand factor. So this Willebrand factor, what it does is it tells, I mean, like it monitors how much of a platelet plug is necessary to maintain the loss of blood. It does not just keep on adding the platelets forever and ever. Okay, so that's another thing. So now coming to coagulation. Now coagulation of blood is a very important, and it's a cascade mechanism. So when I say cascade mechanism, you just have to look at this slide. So in this slide, we see that in one column they have given factors, and in the other column they have given name of the factors, and then what pathway they are involved in. So we'll make it simple so that we understand. So for the coagulation of blood, there are 13 different factors that all of them are necessary to perform or function in 100% so that the complete coagulation is achieved. So if you observe this, factor 1, factor 2, factor 3, factor 4, like that till we factor 13, we have these factors, and they have names also. And some of the very important factors that we have to remember is factor 1, which is a fibrinogen, factor 2, which is prothrombin, and then we have factor 4, which is calcium ions. Remember this, very important. which of the following ions are required for coagulation of blood was the question. So people forget where the calcium comes into role. So factor 4 is calcium ions. Okay. Next comes factor 8, factor 9, and factor 11. So factor 8, factor 9, and factor 11, you're going to study about these factors very detailedly in class 12 for a genetic disorder or clotting disorder. But anyways, so factor 8, 9, 10, factor 8, 9, 11 are called as anti-hemolytic factors. So these anti-hemolytic factors are special proteins like the rest of them. So these factors are also very, very important. Okay. So now we understood that there are 13 factors. All the factors are very, very important to achieve coagulation of blood. So what is happening, students? So there are two different ways in which the clotting mechanism or coagulation mechanism happens. One is called as intrinsic pathway and the other one is called as extrinsic pathway. Okay. Intrinsic pathway is also called as a direct pathway, and the extrinsic, oh, hold on, here it is. Okay. So extrinsic pathway is also called as direct pathway, and intrinsic pathway is called as indirect pathway. So it's very so what is that? So let's talk about extrinsic pathway. Now, what happens once your blood is exposed to these extravascular layers, or when once the blood vessels are ruptured? So that will activate factor 3. So factor 3, what it will do is inactive factor 3 will act. Sorry, factor 3 will activate inactive factor 7 to active factor 7. Nothing. With the injury, what happens? You have your factor 3 formation, and the factor 3, what it will do, it will start activating factor 7 to factor 7a, that is active factor 7a. 7 and 3 combine. What happens when 7 and 3 combine? It becomes factor 10. So factor 10 is also called as common factor. Okay. Factor 10 is also called as common factor. So that is called as extrinsic pathway. Very easy, right? Now look at intrinsic pathway. Now, just because of the exposed to damaged vessels, now what happens is we have the formation of factor 12. Factor 12 is activated into factor 12a. And then with the activation of factor 12, that leads to a series of activation of factor 9 to 9a. Okay. And sorry, factor 11 to 11a, factor 9 to 9a, and then factor 8 to 8a. So once you have activated factor 8, the factor 8 or that will lead to the formation of factor 10, which is a common factor. So both this intrinsic pathway and extrinsic pathway, they lead to the formation of factor 10. Okay. So once you have the factor 10, what happens is a very important step. Once the factor 10 is formed, the factor 2, what is the name of factor 2? Prothrombin. Pro means inactive. So the removal of pro will make it active. Okay. So the prothrombin is converted to thrombin. Excuse me. So that is inactive factor 2 is converted to active factor 2. So they will ask you what is factor 2 in coagulation of blood. So your answer should be prothrombin. So it starts from like this. Okay. Prothrombin, prothrombin, sorry, prothrombin is converted to thrombin. Okay. And once you have thrombin, now this thrombin will activate inactive fibrinogen into active fibrin. So it's just like stitching, people. So this fibrin is like a fiber. Okay. So they become insoluble fiber. So this insoluble fiber, they start knitting together or stitching together to form a very prominent barrier so that the blood loss is prevented. So this is how the coagulation happens. Okay. I know that this was a little bit of more information. That's why I told you. If I tell you that there are 13 factors are involved to perform coagulation, probably would just not understand better. So that's how it happens. See, if any of these factors, whether it is factor 9, factor 11, factor 8, or here it is factor 7 or factor 3, any of these factors go missing. Now what happens here, students? So this will lead to the failure of coagulation. Whenever the blood stops to not clot properly, now you know what happens. It can keep on leaking out of your body, and then you might go under severe loss of blood. So that's why coagulation mechanism is very important. It's a permanent, or it is a very stable pathway. So for that, we require 13 factors. So remember, we have to remember this calcium ions, factor 8, factor 9, factor 11, so and factor 2 and factor 1, students. Okay, very easy, right? Prothrombin to thrombin, fibrinogen to fibrin. That's it. And then calcium ions, antihemolytic factors A, B, and C, which is 8, 9, and 11. Okay, clear? Am I audible? Yes, students, are you following me? Wait. Yeah, perfect. Chalo. So let's go for the next one. So now we have to discuss very interesting fact, very interesting thing, that is our blood typing. So when we look at what happens when a person undergoes a surgery, or when a person met with an accident, or when a person is suffering from a specific disease. So what happens? Can any of us go and donate blood? Yeah, it's not possible, right? What do we tell? So we ask people, what is your blood group? And some of you will tell my blood group is A, some of you will tell my blood group is B, some of you will tell my blood group is AB, and some of you will tell my blood group is O. Sometimes you tell my blood group is AB positive negative, or B positive negative, something like that, right? So what are these blood grouping? If you have to understand blood grouping very clearly, let me give you an example. Now, I'll write a circle here. Consider this as a human body. Now, whatever comes from outside of the human body, which our body cannot recognize, anything it can be. For example, it can be bacteria, it can be viruses, it can be fungi, or any of the allergic things like pollen grains, dust, anything you say. So they're all called as antigens. They're also called as what? They're all called as antigen. So antigen is foreign to us. Now, if I look at the red blood cells, okay, they have plasma membrane, and the plasma membrane of the red blood cells, they produce this a special type of sugar. Okay, yeah, they produce a special type of sugar on the surface of this plasma. Your red blood cells, they can be categorized in. So first, what do you have to understand? What is an antigen? Antigen is something that is from outside the body, which can harm you. Okay. Now, whenever there is an antigen, there is a formation of antibody. So, what is antibody? Antibody are protein produced in our body. Okay. What do these proteins will do? These antibody proteins, they help in fighting these antigens. Okay. Now, why did I tell all these things? Very simple. Okay. Let me show you this. Okay. ABO blood grouping. Here it is. Now look at this, students. If I talk about A blood group, now you can see that the presence of A antigen is there. Now you can ask me a question, sir. If I have A blood group, why are you calling it as an antigen? Correct, right? See, why are you calling it as antigen? Because it is in my own body. If it is in my own body, how are we even calling it as an antigen? We are not supposed to call it as an antigen, right? So what happens is, students, let's say that a person with an A blood group having this antigen A on the surface of the blood, or red blood cells, unknowingly they donate their blood to B type, okay, B blood group. Whenever this happens, right? So if a blood group donates the blood to a B blood group, what happens is there is a reaction called as agglutination. Okay, agglutination. What is agglutination? Now, since the A is very foreign to B, see, my blood group is A and the other person's blood group is B. Now, my body only knows about A, not the B. So it's the same way for the B people. They only know about the B, not the A. So if I mix the blood, what happens is now there is this immune response. What kind of a response? The A blood group, which is donated to the person for a B person, what happens is all the red blood cells starts clumping together, okay, and then it is degenerated. So they start blocking up in the veins, arteries, etc. Slowly, okay, not immediately. It's not a rapid reaction, but what happens is it slowly starts clumping together, which you call it as agglutination reaction. So as it agglutinates now throughout these vessels, now this can be very dangerous. It can lead to the failure of kidneys at excessive amounts. Okay, so that is why the blood group is very important. Okay, so what did I tell you? A blood group person has A antigen, B blood group person has B antigen, AB blood group person has both A antigen as well as B antigen, and the O blood group person has no antigen at all. Are you understanding? Chalo. Now, you might ask me a question. Sir, why did when I donate my blood from A to B, if I donate my blood, which is A blood group, to a B person, why this reaction took place? If you check the plasma of an A blood group or a B blood group or AB blood group, or what we see is we see antibodies, right? So if you see A blood group person, they have anti-B antibodies. If you see B blood group, they have anti-A antibodies. If you see AB blood group, they don't have any antibodies. And that's what makes AB blood group people very special. Okay. And they're lucky people for a reason. If you see O blood group people, they are the poor people. Okay. Very poor people. For there is a reason. Why? Because their blood has both anti-A and anti-B. What you have to accept is, what you have to understand is, if I have a blood group, it will definitely reject B. That's why it is anti-B. If I have B blood group, definitely it will reject an A blood group. That's why it has anti-A. If you see AB blood group, since they have both the antigens, now what happens is, students, they have both the anti-universal acceptors. Whereas if you come to O blood group people, I told you they're unlucky. Why they're unlucky? They have both anti-A. They reject both A blood group as well as B blood group. And these people are only donors, very generous people. They can donate blood to any of the blood groups. They can donate it to A, they can donate it to B, or they can donate it to AB and O. So they are called as universal donors. Why they are called as universal donors? They don't have any antigens. If they don't have any antigens, you can go to A, you can go to B, you can go to AB or O itself. So this is called as blood typing. And how do we check these blood groups? Basically, you can see here agglutinated red blood cells. So we use anti-A, anti-B, anti-D, anti-Sera. So these are some testing kits. When I put anti-A, anti-Sera in A blood group, the blood will clump. If I put anti-B, anti-Sera in B blood group, the blood will clump, okay, or deglutinate. So this is how we will be able to understand what blood group is what. So that's why blood grouping has become one of the key discoveries in saving millions of lives and simplifying the donation of blood during the needy times. Okay, did you guys understand this blood typing? Yes, students, can I go further? Yeah, thank you for your response. Okay, the next one is RH factor. So what is RH factor? RH stands for rhesus factor. So basically, this is, see, if you ask me, sir, is it only A antigen, B antigen are only present? You are wrong actually. There are more than 20 different antigens in our blood. Okay, so one of the most problematic antigens are A antigen, B antigen, and RH antigen. Now we finished with A and B. Now it is RH. What is RH stands for? RH stands for rhesus factor. Why it is called as rhesus factor? Because it was discovered in a monkey called as rhesus macaque, and its blood is very similar to humans, and that's how the discovery of this antigen took place. So now, whenever we use anti-D, anti-Sera, we can detect RH. Whoever has RH antigen, they are called as RH positive, and whoever don't have RH antigen, they are called as RH negative. So it doesn't matter whether you have A blood group, B blood group, O blood group, but the key importance comes with whether you are RH positive or RH negative in certain cases. That's how you get AB positive, A positive, B positive, A negative, right? So that's how you are grouped. Now, why we have to do RH grouping? So now sometimes what happens, the RH problem or the problem of the RH factor comes in the during the stage of a pregnancy. So this is called as erythroblastosis fetalis. So erythroblastosis fetalis is a condition. So another name for this condition is also called as hemolytic disease of a newborn, or HDN disease. Let's see what happens here. Now, look at this. If you see the womb of mother, womb where the baby is developing, right? So we have the uterus or the womb, what we call it as. So this is the uterus, let's say. Okay. Now, this is how the fallopian tubes and everything are connected. But anyways, this is a female reproductive system, very rough representative of female reproductive system. Anyways, so here, students, you can see that what you can see is there is a functional body that connects to the baby. Okay. And this is the cord, correct, right? Umbilical cord. And this is the baby. Okay. So this is the baby. I know this is very, very rough. So this thing is called as a placenta. Okay, the functional body, which is a barrier between the mother and the uh, baby, is called as a placenta. It has many functions to play. Okay, it has very important roles to play. Now, when does RH becomes a problem? Now, see here, RH positive blood, RH positive blood, when it is mixed with RH negative individual. Okay, now what happens, they undergo this hemolytic disorder. How does it happen? Now look here, this is the this is the placenta, right? This is the infant blood. This is the infant means baby, infant blood. And this is the mother blood. So this is the mother tissue, and whatever the finger that is there inside, that is the baby's blood. Now sorry, yeah, excuse me, 1 second. Mm, 1 second, 1 second, students. Yeah. Um. Yeah. Sorry. There is a small error. Whenever there is RH negative, yeah, RH, obviously whatever it said is right. So let's say that mother's blood is. Remember this. Mother's blood is. Mother blood is RH negative. Okay. And baby is RH positive. Okay. Now what happens here? Since baby is RH positive and mother is RH negative, in the first exposure, what mother will do is mother's blood will think that baby's blood is enemy or it is foreign. Why? Because baby is positive, right? So it has antigen. So it starts producing antibodies against it. See this Y colors, Y symbols are antibodies that are sent against the baby's blood to fight. So when these antibodies are produced in excessive amounts, they start attacking the baby's blood, so that the blood starts degglutinating and it starts forming clumps in the infant's blood vessels. So that will lead to a hemolytic blood disorder in the infants. So what we have to remember is, when does this condition happens? The condition occurs when mother has RH negative blood and the baby has RH positive blood. This is the key thing that you have to keep it in your mind. Second thing, what happens here? When mother is RH negative and the baby is RH positive, the mother who do not have this RH antigen, once she gets introduced to baby's blood, the body of the mother starts thinking that the RH positive blood of the baby is enemy or an antigen, because positive means what, they have RH antigens. So this mother's antibody, they start targeting this infant blood. So these blood cells, they start clumping together. So once they start clumping together, that will lead to deglutination reaction, and finally, it leads to small clots throughout the blood vessels of the baby. Now this can lead to the malfunction in the liver, failure in the kidneys, etc. But there is one small change. What is that? Let's say that during the first pregnancy, even though the mother is RH negative and the baby is RH positive, the effect is very less. The effect is very very well integrated. So that's why it prevents, as the number of pregnancies increase. Now what happens is the barrier between the placenta and the baby is reduced, or it becomes lesser and lesser as the number of pregnancies happen. So as this occurs now in the second exposure or in the third pregnancy, that will lead to severity of the baby's development. So in most cases, the baby loses its life, or it has the organ failure. Students, on with the next topic, and please feel free to ask me questions. So the most interesting part, we understood many things about body fluids. We understood the composition of blood. We understood the composition of plasma, and what are the key characteristics of the formed elements, that is red blood cells, white blood cells, platelets, how are they formed, where they are formed, etc. And other characteristics like blood grouping and RH factors we studied. And the key organ to just supply the blood to all the tissues in our body, whether it is from brain to our toes, to kidneys and our lungs, and draining the nutrients from our intestines to delivering it to the every tissue. So it is done by our human heart, such a wonderful machine. So some of the key facts that we have to understand about human heart is, human heart, it beats around 75 times in a minute, okay, and it pumps around 70 ml of blood per contraction. Every time it contracts, it pumps around 70 ml of blood out of the heart, and for every minute we supply around 5.75 liters of blood across the body. Now, coming to the position of the heart is present in the thoracic chamber. Now, you can see that in the left lungs and the right lungs, there is a small dent like this. So, this is called as left cardiac notch and the right cardiac notch. Between the left cardiac notch and the right cardiac notch is a space which we call it as car. Oh, hold on. We have a message. Yeah, there is a space, and this space. For 1 second, students. I thought there is a. Okay. Yeah. See here, this space, this exact position where the heart. Oh, excuse me, students. Excuse me. I'm really sorry about that. Okay. So let's get back to where we stopped. Yeah, the position of the heart. Yeah, when you look at the left cardiac notch and the right cardiac notch, here there is a space that is specially designed for the heart to be positioned. This position we call it as mediastinum. So if you just start counting the number of ribs from the top, when you come to the fifth rib and towards the center is where you have the heart is positioned. Actually, heart is at the center, but it tilts a little bit towards the left side. Okay, so many people confuse that the heart is on the left side. No, it is centrally located in the thoracic cavity, and you have to remember this name called as mediastinum. So mediastinum is a place where, see, you can see here, this entire region is where the functional heart is located. Okay, so this is the anatomical position. Now coming to somebody who has undergone a cardiac arrest, so which we generally refer it to heart attacks. Okay, and medically, which is also called as myocardial infarction. See, whenever a person goes into a cardiac arrest, so what we do is we perform something called as CPR. What is that? Cardiopulmonary resurrection. So what is cardiopulmonary? Cardio is heart, pulmonary is lungs. Resurrection is you pump the blood from the heart to the lungs so that your restart. Your heart is restarted. So there is a mistake that many people do. Whenever you have to pump the heart, you have to place your hand very perfectly around the fifth rib at the center, and then gently press several times. In a minute, as fast as possible. Uh, Maybe 50 to 70 or 100 compressions as much as possible. Now there is a possibility of the heart being, restarted. In case if you place your hands on the wrong position and if you do it very with a high force, you might end up breaking the bones, especially the rib bones, and it might puncture the lungs, and the chances of saving a person might go in the opposite direction. You might really kill a person if you don't really know where to place your palm and then resurrect a person. So here is a picture that gives you a very good idea about where to place your hands for the cardiopulmonary resurrection. That is the T4 and the T9. That is thoracic vertebrae 4 to thoracic vertebrae 9 is your go-to place for the cardiopulmonary resurrection to be practiced. Okay, it's a life-saving. What do you tell? It's a life-saving fact because it really happened once when a person's heart actually, what do you tell? He went under myocardial infarction or a heart attack, and then literally there was a flat line in his ECG, and eventually what happened is his heart got resurrected while performing the CPR. Okay, so coming to the anatomical dimensions of the heart is around 12 centimeters in length and 8 centimeters in wide and 6 centimeters in thickness. So the female heart is quite lighter. It's around 300 grams or 250 to 300 grams, whereas male heart is around 300 350 grams. Okay, so coming to the anatomy of the heart. So let's get back to, hold on. If I have a better picture. Yeah. So we know that our heart is divided into four chambers called as receiving chambers, and the lower chambers are called as pumping chambers. So the upper chambers are called as right atrium and the left atrium. You can see here. So the right atrium and the left atrium, they act as the receiving chambers of the blood. So what happens is the blood from the superior vena cava and the inferior vena cava. Superior vena cava is a blood vessel that actually brings the deoxygenated blood from upper body. Okay, I'll write it here. Upper body. So it brings it from upper body. 1 second, students. Are you able to hear me, guys? Yeah. Is my voice clear?